Hello, everyone, and welcome to today's webinar titled Looking for Evidence-Based Oncology. Do Targeted Proteomics. This webinar is part of the Cancer Research and Oncology virtual event. And I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Luminex. For more information about Luminex, go to www.luminexcorp.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, just click on that Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers continuing education credits. Click on the Continuing Education Credits tab located on the top right-hand corner of your presentation window and follow the process to obtain those credits. Without further ado, I now present today's speaker, Dr. Christian Regenbrecht. He is the CEO of Cell Phenomics and the CEO of ASC Oncology. For a complete biography of our speaker, please visit the Biography tab at the top of your screen. Dr. Regenbrecht, welcome. You may now begin your presentation, sir.
Thank you very much for the kind introduction and the invitation to present today. So also thank you to all the listeners and viewers in the times of Corona that you're still interested in cancer, because nevertheless, cancer is still an important disease lurking around the corner. So today I'm talking about our approach to personalized oncology. And what was fueling it is an interview by James Watson in the New York Times 2016. James Watson said, locating the genes that cause cancer has been remarkably unhelpful. The belief that sequencing your DNA is going to extend your life is a cruel illusion. And I totally agree with it. Today, politicians, doctors, the broader audience, they also always see personalized medicine as sequencing the cancer genome. And why this has been unhelpful in the past is very well summarized in a paper from 2017 in Cancer Research Reviews by Amy Brooks. And let me just talk you through it. So our mental cartoon is that we have a cancer where a normal cell receives a mutation or a set of mutations, develops a new cancer phenotype, and this new cancer phenotype could be investigated as a mechanistical role in the signaling pathways of the cells. For example, with the RAS pathway, it's a very well-studied pathway. Each and every player was known and understood. So the consequence of therapy was that people started developing targeted therapies against the aberrant proteins. And the idea was, if the protein is targeted well, then the cell should go into apoptosis. But the biological reality is much more vague because we have plasticity here, heterogeneity, meaning that within the same tumor, there are different clones existing in there, but also on the same genomic background, just in the context of tissue, you have more resistant cells and more sensitive cells. And this is a dynamic process. And with sequencing, for example, you only see a snapshot. You can only see what is happening at a certain time. You cannot follow this biological dynamics. So based on that, this means that targeted therapies have cells that are going into apoptosis and are targeted well. And you have surviving cells that are stress-induced cancer stem cells. And this also explains why second or third line treatment usually is less beneficial to the patient than a first line treatment. So cancer has various genomic mutations underlying it. And the treatment is more or less the same depending on where the cancer is and which grade and stage it is. And it is only now that we start to understand that basket trials based on the same underlying molecular events are needed more eventually than the location and the site of the tumor. And that means that in average, for solid cancers, about 50% of the patients benefit from the first-line therapy, only 12.5% from the second-line therapy, and from third-line therapy, 3.75%. So this is just an estimate, but it shows very well the earlier a patient receives the right treatment, the better it is. So we are not going to design new drugs, but our ultimate goal is to use the arsenal of weapons that we have for fighting the war on cancer. We want to bring the right drug to the right patient at the right time. And how we do that? So we use cell culture models that we derive freshly from surgical specimen that we receive. And there are currently like two main areas in the field of 3D cell cultures. One is mechanical force models, so-called spheroids. That is like hanging drop technology or ultra-low attachment plates, where the cells have to grow together just by the sheer forces of the droplet. And I always compare that to cantaloupes grown in cubic glasses. This is not the biology of a melon. This is forced just by the surrounding. What we and other groups are focusing on is 3D scaffold, so-called organoid models. And here we have the three-dimensionality and the mechanical properties with the chemical complexity, which ultimately leads to a native organization. And this degree of organization is so good that it really mimics the original tumor, hence tumor organoid. What you see here is on the left side, a spheroid, on the right side, an organoid, both derived from the same patient tissue. And I think you can easily see that this looks more like a mosaic. And this, on the right side, the organoid, has a much more structured domain-like appearance. And how good is this in reality? What you see here in the top row is tumor tissue from colorectal cancer, as used by the pathologist for determining the diagnosis. And here is the 3D cell culture derived from the original tissue. And even if you're not a pathologist, I think it's easy and convincing to see that these patterns look very much like the pattern of the upper row. So that is how we see it. If you look at such an organoid under the microscope, you see a structure which first of all seems like a lump of tissue. But if we do stainings with proteins like ezrin or F-actin, do an overlay, you see this tube-like structure going through this organoid. In the next slide, I'm trying to present to you, if the video is running, yep, how a 3D Z-stack works through such an colon organoid. And you can see like crypt-like structures here and villi-like structures here going into that. So this is a highly ordered structure where a pathologist would be able to tell you, okay, this looks like a colon cancer. In fact, we did not only do that for colon cancers, but we were able to do that also for breast cancers. Did a little blinded experiment where we derived from 5 different patients, 5 different organoid cultures, and sent it back to the pathologist, and he was able to map the right model to the right patient just based on morphology, expression of markers, and so on. So we have reliable, faithfully recreating models recreating the original donor tissue. So from that, how do we get closer to the patient? We do cytotox screenings with those models. For this, together with colleagues from Eli and Lilly, we have developed a platform for screening 3D organoid cultures. In brief, we pre-culture the organoids, disintegrate them, and then we use a liquid handling station to use the single suspension and put it in a 384 well plate. Then we add the growth medium to the plate. And while the cells recover from this procedure, we do an automated serial compound dilution on a compound plate. And basically, we then stamp the compound dilution plate on the assay plate. And then we test for usually around 4 days the cells. Then we put cell titer glow on them. Cell titer glow is an ATP-dependent assay. And from that, we determine the viability of cells. This procedure has been deemed a best practice approach, and we're quite happy that it has been acknowledged as the way to do it. And just to show you one of our first experiments that we did, this was to establish the assay. We used staurosporine, a pump kinase inhibitor, just to show the concentration dependency. So we have here the relative luminescence units. We have each single data point, concentration of staurosporine. And if we then calculate from that the percent inhibition, we see this nice sigmoid curves representing the drug treatment and the IC50 here around the 50% bar. We can also screen that in a time-lapse video. And what you can see here is an untreated organoid growing. Here again, we used staurosporine on different concentrations, and it's easy to see over time how growth is really inhibited by the drug in a concentration-dependent manner. So now we have a good model. We have a way to interrogate the model. How do we get close to the patient? So the idea is each drug has a Cmax, a maximum concentration in human blood. And this is specific to each drug. And so we found out which are the Cmax values from investigator brochures, ASCO posters, etc., publications. For some of those drugs, I have depicted it here. Then we have that in milligram per liter, which is unfavorable to use in the cell culture. So we needed the molecular mass, and we could recalculate it into micromolar units, something that would suit us very much in the lab. So with these values, we know this is the maximum amount of drug that can be sold in human serum, which is the maximum amount that theoretically could reach the tumor, meaning this is our upper bar for the drug where it has to reach an IC50 to reach at least stable disease, meaning if we kill 50% of the cells and the other cells double in the same time, we have stable disease. So the lower the concentration where we reach the IC50 is, the better is the predicted outcome for the patient. Let me show to you some practical examples of that. First of all, there is peritoneal metastases of colorectal cancer. The clinical situation is dire. It is a variant of colorectal cancer metastases, and it is for about 15% of the colorectal cancer patients, they develop peritoneal metastases, and it's associated with a poor prognosis. Surgery is long, difficult, and for the patient, not so easy to take. It's a complete peritonectomy. In terms of chemotherapy, there is no guideline protocol for this tumor entity, meaning people try 5-FU, irinotecan, oxaliplatin, etc., etc. Some larger hospitals also use HIPAC, but HIPAC is associated with high morbidity, definitely not standard of care. So the clinical needs for cancers like that is, can we find markers for patient stratification, or can we identify new targets for an improved therapy? And what we have done here is each diagram represents a specific drug. So 5-FU, oxaliplatin, docetaxel, SN38 is irinotecan, and so on. And each graph in these diagrams represents a single patient. And you can easily see that, for example, here the yellow patient, or the brownish line, is the best responder because it's on the far left side. But for oxaliplatin, it is the worst responder because it doesn't even reach the IC50 level, so we cannot even kill half of the cells. And if we go through that, you can see that basically for each and every patient, we have to find an individual drug or drug combination that may be useful. And even those two compounds, companizib and LY29402, work in the same molecular pathway, but on different targets. And you can see that even there, patients respond differently. So it is important for us to know how the targets are activated and where the phosphorylation occurs. And these are things that we cannot learn from sequencing alone, because that's post-translational modifications, and you don't see that via RNA-seq and so on. So this is just another representation of the data I've just shown to you. And it shows you how wide the data spread per model. And you can easily identify the best responders and the worst non-responders. And that gives us a good idea and a good way to identify those models that we want to investigate further by taking just the top 3 or the lower 3 and test it. So that is basically our setup for the cell cultures. And now we add another layer of complexity to it, because as I mentioned before in the introduction, tumors are heterogeneous. And so what we call reverse clinical engineering is trying to modeling the intratumor heterogeneity. What does a tumor look like? I think most of you know we have the one original cell where the tumor starts from, and from there different clones can develop, and the chances increase for multiple clones with each and every cell division. So with billions of cell divisions in any metastatic cancer, we have a variety of clones in there. So what we do is, for example, here from a colorectal cancer again, we took a heterogeneous tumor, received from the pathologist 5 distinct regions. We cultivated each and every region of this tumor and did a drug screening. And don't worry, I don't go through all of the graphs now, but what I want to show you is, for example, here in AZD8931, there's an up to 30-fold difference between the different regions of the same tumor at the same time. And for me, this was a eureka moment. It was a eureka moment because I always ask myself, why in the 21st century, the first-line treatment in colorectal cancer is still a systemic chemotherapy? And now I understand that. It's due to the intratumor heterogeneity. With the systemic chemotherapy, you reduce the amount of clones in the tumor, making it more likely that on the fewer surviving clones, the drug really works. And I think that is an enormous understanding, at least it was for me, in how to treat and handle those tumors. So what we did then next was we did a transduction, lentiviral transduction with the resistant and the susceptible population in green and red. We mixed them and injected them into animals. So what we see here is we started the treatment, and here's the treatment stop about 46 days after implantation, which is quite a long time. And you see that with everolimus, the control, and pictilizib, we had to sacrifice the animals because the tumors were growing very quickly. But with all the other drugs, we were able to keep the tumor burden in chest. So, and then we stopped the treatment and did a long follow-up. And what you can see here is that each single drug or combination, the tumors grew back, except for the combination cetuximab plus regorafenib. And that was very interesting because the patient was RAS mutated, and cetuximab has only an approval for wild-type RAS patients in colorectal cancer. So when we tried to publish this data, we were asked by reviewer 3, hey, why do you try cetuximab at all? The patient would never receive it in the clinic. And the answer to that is quite simple. We tried it because we can. We have the ability to screen in a 384-well format in a way that we can try out things without putting the patient's life and health at risk. And as you see here, cetuximab alone didn't work as expected, but neither did regorafenib. Only the combination of these two was highly effective in this patient. And I think this is the approach that we want to go. I was asked where I wanted to present, and I said I want to present in the disparity session. Why was that? Because I think every cancer patient is a disparity when compared to a large cohort, and we have to go back to the focus of the individual. So the next thing that we then did is we took our PDX tumors, did some labeling with them. We found the GFP and Sherry cells, we merged them, and what we saw is they existed side by side. They existed not intermixed, but side by side as regions. And that is, again, very much closely to the organoids as compared to the spheroids. We did some faxing, and we found that there were tumors with a low M-Cherry population compared to a high M-Cherry population, and we tried to map that. And this is a quick representation of what we found. In the control animals, about 80% of the cells were green and 20% red. The tumors were shrinking a little bit when treating with trametinib, but green cells became more favorable. Cetuximab alone basically did not shrink the tumor, but still the selection was a little bit preferring the green cells. But for example, when treating with trametinib plus cetuximab, tumors got very small and consisted of about 75% red cells. What does that mean? That means from a clinical perspective, the tumor was shrinking. One would suggest the treatment is working, but what we saw is it was selecting for the more aggressive population, the red one. And with the more aggressive population, this easily explains why second or third line treatment will be harder to have positive results for the patients. So that is, I think, a very nice experiment to show the biology of these things. And now we add a third omics layer, and it's a targeted proteomics approach called GGWest, developed by our friends and colleagues at the NMI. And this is based on Luminex technology, which I think is quite elegant. So what does GGWest do? You just do a classical Western blot. You do your SDS page, you plot it on the membrane, then you cut out the lanes and the bands. You elute the proteins and couple them to Luminex beads. Luminex beads are fluorescence beads with specific color coding, and that helps us to do multiplexing, because we can now pool them and know which color is associated with which molecular weight. And that helps us to use the amount that you usually need for a single Western blot to do up to 800 analytes from one sample. In terms of the organoids, that means from 4 wells of a 24-well plate, we are now able to do up to 200 proteins and phosphoproteins. And this is, for me, the coolest thing about this technology. We see the post-translational modification. We see on the proteomic level, is the pathway activated or not? And this is such a benefit for what we are doing that I cannot stress it enough. So we see here, just to show you, these are sarcoma samples that we have used, and these look distinctive from other cell types. So as control, we put mouse liver in there. And the system is so nice that, in fact, for the tumor with the red and green cell populations I showed you before, we could explain why certain drugs could not work and why other drugs had to work. So if we look at the PI3 kinase mTOR axis, we see there is basically no different expression or activation from the surface down to the transcription factors. So the AKT is differently expressed, but on the phosphorylation level, it's the same. AKT2, the same, and so on, going downstream one single experiment. So all in parallel, all from the same sample. If we switch over to the MAP kinase pathway, we see the receptor level, no difference. We see on the RAS level, no significant difference. BRAF phosphorylation, no difference. MEK PAN expression, no difference, but then we see the differentiated phosphorylation at MEK1/2, following down to ERK, P90, RSK, so we could see that. And this is linked to an activating mutation upstream of MEK1/2. So with the genomics, we found the activating mutation. With the targeted proteomics, we could explain it. And I think this is so awesome for future generation of patients that this will help very much. Sorry, this slide is in German, but so it starts with patient-informed consent. Then biopsy is taken. Then we have the classical clinical pathology with histopathology, immune histology, molecular pathology, FISH, sequencing, chromosomal operations, so the standard of care. On the right side, we have what ASC has to offer. We do the patient-derived 3D cell culture models. We do the in vitro pharmacology, generate a pharmacological profile. In parallel, we have the targeted proteomics, integrate that. And we really discuss with the oncologist in the tumor boards our data and compete with the genomic data for the best solution for the patients. So, because I think it's very important to see in which context this works, sarcomas are basically the orphans of oncology. So sarcomas are soft tissue and bone tumors, and very understudied. And we use them in the cell culture, and we have now an average take rate of 73% as of August 1st. And with our new method of cultivating sarcomas, you see, for example, some subtypes of these sarcomas, we have up to 100% take rate, meaning a tumor entity which is totally aggressive, that usually hits teenagers, young adults, harsh chemotherapy. We have now a way to provide those people with crash test dummies, where we can pre-test which treatment will help or will not help. Here is just some slide to show how close we are to the situation. The Vimentin is positive. All the others negative. Desmin, we find single cell in the patients, positive, negative in the cell culture. Ki67 for proliferation, in the same range of those Ki67 expressions. So we are very, very close to the clinical situation here. Different type of sarcoma, all the cohort of markers that we have tested, again here, Ki67, 80% versus 80 to 90%. So this is really close to the patient situation. And if we look at the data from the drug screening, we can easily see for those tumors which drugs are favorable and which are not. We have drugs where we have no response at all, where we have just a flat line, not a curve. But we have drugs that are so effective, in this case, staurosporin as a pan-kinase inhibitor, control, cannot be used clinically, just to show you that is an effective drug. But actinomycin D, for example, nice curve. We reach very early in IC50, and this is something to suggest to the patients. Or here, doxorubicin, even at the lowest concentration we administer to the cell culture, already below IC50. And this is, from our perspective, very much helping the oncologist to choose the right drug within the framework of his guidelines, and also help the patient not only to accept and comply to the treatment, but also in the course of treatment, many patients have told us, if we wouldn't have seen the data black and white, we wouldn't have gone through this pain of treatment. And here other sarcoma patient, different molecular profile. You see here, for example, doxorubicin is still working, but not as good as with the others. But trabectedin, in this case for the patient, works like a charm. So these are the things that we are working on, and where we think that we bring ahead the oncology, and we shape the oncology of the future. And of course, I have always the pleasure of presenting this data and being happy to show that to you. But as you can see here on that screen, there's a large team behind it at the company. Everybody was working hard, specifically for the patient projects. We have full support from companies like Luminex that help us investigate these things. Thank you very much for that. And the video that you see here is not a computer animation, but this is some scanning electron microscopy video. Basically, it's a stop motion animation. We did a photo under the SEM, moved the stage, took the next photo, and so on. And what you see is an organoid of a colorectal cancer, just to show you that there's also a kind of beauty in these tumors. Thank you very much. And I hope I find some questions now if I move back to the presenter stack. Thank you.
And thank you, Dr. Regenbrecht, for that informative presentation. We will now start the Q&A portion of the webinar. And if you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. We have quite a lot of questions already coming in. Our first question is, how do oncologists respond to your approach?
Well, that totally depends. We have oncologists in university hospitals that say, okay, we know all the stuff and we know it better, and we have our experience, and we don't want anybody to guide us through our decision-making process. But those people are rare. Most people are happy if we find a way to support them in the decision-making process. And specifically, people working in peripheral clinics or their own doctor's office are happy that they're able to offer their patients these kind of services, because that was unimaginable a couple of years ago. That was top-notch comprehensive cancer center level treatment and diagnosis.
Thank you so much. Now, is it true that a person who has cancer more than other people are in the high risk of COVID?
Oh, interesting question. I'm not a physician, but you can easily imagine that with chemotherapy and all the side effects, your immune response is low. And with that, the risk of catching COVID is very much higher than with a normal healthy person. So definitely risk group.
Thank you so much. And I want to thank our audience members for these great questions coming in. Now, do you think that CRISPR will play a key role in the fight against cancer?
Well, it will play a key role in terms of research, because if you can CRISPR in specific mutations or CRISPR them out, it helps enormously to understand the biology, to identify biomarkers. I think we are very far away still from using it in the human body and really treating the patient with a CRISPR-based approach. But for the research purposes, I think it's boosting cancer research. Yes, definitely.
Thank you, Dr. Regenbrecht. Now let's talk about sarcoma. We have an audience member that wants to know what causes sarcoma, and is it inherited or is it acquired at some point in life? And if it is inherited, what can cause it? Well,
sarcomas are, as I said, rare tumors. It's less than 10% of all solid tumors. Patients often report that at the site where the sarcoma was diagnosed, they had a broken bone or any other injury. But it is more a felt knowledge, because large studies have shown that there is no correlation. In fact, the genomics of sarcomas looks very different than for carcinomas. In sarcomas, you either have very few mutations, or the genome looks like you have aimed at it with a shotgun and it's totally corrupt. So it is definitely a genomic disease. I do not see any direct environmental elements that play into the role. It's spontaneous mutations and, of course, inherited by the predisposition with those tumor genes. So often it's fusion genes that you find in sarcomas more than you would expect, for example, in carcinomas.
Thank you so much, Dr. Regenbrecht. Our next question, will you need to do some clinical trials to validate your method of treatment?
Yes, and we are currently doing so. We are currently designing a phase 4 clinical trial for sarcomas, even for first-line treatment patients. And within the German health system, we are currently also working together with university hospitals to conduct clinical trials on various tumor entities, together with the tumor boards and for advanced cancer patients in third-line treatment that are to today's standard of care beyond care.
Thank you so much, Dr. Regenbrecht. We have time for a couple of more questions. Let's talk about your partners. Do you already have clinical partners in place?
Yes, as I just mentioned, yes, we have some university hospitals in Germany we are currently working with, but also with clinics in Austria and the UK. We work quite closely and exchange knowledge and patient tissue. That are those regular partners. And as I mentioned earlier, that small hospitals every now and then want to send samples of their patients to us if they don't find a standard of care treatment that really helps the patient. Yes, so we are currently expanding internationally.
Oh, thank you very much. And actually, that is our last question that we have time for. An audience member wants to know, do you offer the services internationally? So maybe you can expand a little bit on that.
Yeah, so the method that we use allows us to cultivate tumor tissue that needs to be relatively fresh, meaning with everything within 36 hours from surgery to our labs, we can process, meaning the boundaries are more customs than the transportation process. And within the EU, that is totally no problem. It sometimes, especially now in the times of COVID, gets difficult if customs is involved and they don't process the tissue quickly enough.
Thank you so much. And actually, we have one more question that just came in, and I think we have some time for it. What are the challenges to design your clinical trial, Dr. Regenbrecht?
The biggest challenge is, what is the control if you work on a personalized, individualized basis? Who is the control for the patient that we are treating? We don't have twins that we could use. So the problem is the way oncologists and the system thinks, it's in large cohorts. And large cohorts just predict an average risk. But you will not find any oncologist that would put a specific patient's personal risk on such a graph. So that is something where we need to change the way of thinking and to promote the confidence. And that has been done, for example, by us and other groups. There's a nice paper from Vlachogiannis in 2018 where they did a co-clinical trial, and with colorectal cancer patients, in 100% they predicted correctly which drugs will not work, and in 88% they predicted correctly which standard of care drugs will work. And I think that is an enormous step ahead, and that was the first step opening the minds for clinical trials and being a little bit more futuristic.
Dr. Regenbrecht, thank you again for your presentation today and for your time with us. Would you like to provide some closing remarks for our audience before we go?
Yeah, so as I said, thank you very much for the interest, but most importantly, if any of you would like to collaborate with us, we are happy to do so. If you have patients that you would like to support in the decision-making process, we are glad if we can help. And every clinic that would like to run a clinical trial with us, also just approach us. The email address, I think, is in the CV.
Dr. Regenbrecht, thank you again for your time today and for your important research. And before we go, I want to remind the audience that today's questions, any questions not answered today, and those submitted on the demand period will be addressed via speaker and the contact information you provided during registration. Thank you for joining us. And I'd also like to thank our sponsors, Luminix, for sponsoring today's webinar. This webcast can be viewed on demand for 6 months till April of 2021. LabRoots will alert you via email when it's available for replay. And we encourage you to share this email with your colleagues who may have missed today's live event. Until next time, take care, stay healthy. Bye-bye.
Bye.